Welcome to this video on gene expression. Gene expression is the process by which information in DNA is used to produce proteins. Not all genes are active all the time, so cells must control which genes are expressed and when. Cause context and A-level specification requirements from gene to protein, where control occurs. The control of gene expression builds knowledge on DNA structure, transcription, translation, and protein synthesis. It explains how cells with identical genomes can develop into highly specialized cell types through selective gene expression, and that regulation of transcription and translation enables cells to control their metabolic activities, respond to internal and external stimuli, and develop specialized functions within tissues and organs. It integrates molecular biology with whole organism outcomes, linking genotype to phenotype through regulated protein production. Key specification areas include gene mutations and their effects on protein structure, regulation of transcription by transcription factors, epigenetic mechanisms such as DNA methylation and histone modification, and the role of RNA interference. Students are also expected to understand stem cell differentiation, oncogenes, and tumor suppressor genes in cancer development, and the use of genome projects and modern gene technologies. Emphasis is placed on data interpretation, requiring students to analyze gene expression data from techniques such as microarrays and RNA analysis, and to evaluate evidence linking gene expression to disease. Gene expression refers to the process by which information encoded in DNA is used to synthesize functional proteins or RNA molecules, and it involves two main stages, transcription and translation. Control of gene expression can occur at multiple points along this pathway, but in eukaryotic cells the most significant regulatory step is transcription. During transcription, RNA polymerase binds to promoter regions of genes and synthesizes messenger RNA, mRNA. Whether transcription occurs depends on the presence of transcription factors, which may act as activators or repressors. Following transcription, further control occurs through RNA processing, including splicing to remove introns, which can result in different proteins being produced from the same gene via alternative splicing. Translation at the ribosome can also be regulated, for example by controlling mRNA stability or availability. Types of gene mutations and their origins. How mutations alter protein structure and function. Gene mutations are changes in the base sequence of DNA. Mutations can arise spontaneously during DNA replication due to errors made by DNA polymerase, or they can be induced by mutagenic agents such as ionizing radiation, ultraviolet light, or chemical mutagens found in tobacco smoke. The specification requires students to know several mutation types, including substitution, insertion, deletion, inversion, duplication, and translocation. Substitution mutations involve one base being replaced by another and may be silent due to the degenerate nature of the genetic code, meaning the same amino acid is still coded for. Insertions and deletions are particularly significant because they often cause frame shift mutations, altering the reading frame of codons downstream and usually resulting in a non-functional protein. Inversions involve a section of DNA being reversed, while duplications repeat sections of DNA, and translocations move DNA segments between chromosomes. Mutations can be neutral, beneficial, or harmful depending on their effect on protein structure and organism survival. For example, a substitution mutation in the CFTR gene can cause cystic fibrosis, whereas other mutations may confer advantages such as antibiotic resistance in bacteria. The impact of a gene mutation ultimately depends on how it alters the structure and function of the protein encoded. Proteins rely on their primary structure, the sequence of amino acids, to fold into specific secondary and tertiary structures that determine function. A substitution mutation may change a single amino acid, which can have minimal effect if the amino acid has similar properties, or a major effect if it disrupts bonding within the protein. Frame shift mutations caused by insertions or deletions typically have more severe consequences because every codon downstream is altered, often introducing premature stop codons. This usually results in truncated, non-functional proteins. Real-world examples strengthen this understanding, such as sickle cell anemia, where a single substitution in the hemoglobin gene leads to altered protein structure and red blood cell shape. In contrast, some mutations may affect regulatory regions rather than coding sequences, altering when or how much protein is produced rather than its structure. Transcription factors, promoters, and enhancers, epigenetic regulation, DNA methylation, and histones. Regulation of transcription is the primary mechanism by which eukaryotic cells control gene expression. Transcription factors are proteins that bind to specific DNA sequences and either promote or inhibit transcription. Activator transcription factors bind to enhancer regions and increase the likelihood that RNA polymerase will bind to the promoter, while repressors reduce transcription by blocking binding or altering chromatin structure. Promoters are DNA sequences located upstream of genes that serve as binding sites for RNA polymerase. The presence and combination of transcription factors determine whether a gene is switched on or off in a particular cell. This allows for precise spatial and temporal control of gene expression, essential for development and response to environmental changes. For example, genes involved in cell division may be activated only at specific stages of the cell cycle. Epigenetics refers to heritable changes in gene expression that do not involve alterations to the DNA base sequence. One major epigenetic mechanism is DNA methylation, where methyl groups are added to cytosine bases, particularly in promoter regions. Increased methylation generally reduces transcription by preventing transcription factor binding. Histone modification is another crucial mechanism. DNA is wrapped around histone proteins, and chemical modifications such as acetylation can alter how tightly DNA is wound. Histone acetylation typically loosens chromatin structure, increasing transcription, while decetylation tightens it and reduces gene expression. Epigenetic changes play essential roles in cell differentiation, allowing cells to maintain specialized functions over many divisions. They also provide a mechanism by which environmental factors, such as diet or stress, can influence phenotype. Cell differentiation and the role of stem cells, RNA interference, siRNA and mRNA mechanisms. Cell differentiation is the process by which unspecialized cells become specialized in structure and function, and it is driven by changes in gene expression rather than changes in DNA sequence. Totipotent cells, found in early embryos, can give rise to all cell types including extraembryonic tissues. Pluripotent stem cells, such as embryonic stem cells, can form most body cell types but not placental tissue. Adult stem cells are usually multipotent, restricted to forming related cell types. Differentiation occurs because specific genes are switched on while others are permanently switched off through transcriptional and epigenetic mechanisms. For example, muscle cells express genes for contractile proteins like actin and myosin, whereas neurons express genes involved in neurotransmitter release. The specification also addresses the therapeutic potential and ethical considerations of stem cells, including their use in regenerative medicine. RNA interference, RNRE, is a post-transcriptional mechanism that regulates gene expression by preventing translation of specific mRNA molecules. These short RNA molecules bind to complementary sequences on target mRNA, forming double-stranded RNA that either triggers mRNA degradation or blocks ribosome attachment. This effectively silences gene expression without altering DNA. RNRE plays a vital role in normal cellular regulation, development, and defense against viruses. It also has significant medical potential, for example in targeting overexpressed genes in cancer cells. Oncogenes, tumor suppressors, and cancer development, genome projects, gene technologies, and data analysis. Cancer is fundamentally a disease of disrupted gene expression and cell cycle control. Oncogenes are mutated forms of genes that normally stimulate cell division. When permanently activated, they lead to uncontrolled proliferation. Tumor suppressor genes, such as p53, normally inhibit cell division or trigger apoptosis, and loss-of-function mutations remove these controls. Cancer typically develops through the accumulation of multiple mutations affecting different regulatory pathways. Epigenetic changes can also contribute, for example by silencing tumor suppressor genes through DNA methylation. Genome projects and gene technologies demonstrate how understanding gene expression has practical applications. Genome projects involve sequencing and analyzing the complete DNA of organisms to identify genes, regulatory sequences, and evolutionary relationships. Applications include identifying disease-associated genes and developing targeted therapies. Gene technologies such as PCR allow amplification of specific DNA sequences, while sequencing determines base order. Modern techniques like CRISPR-Cas9 enable precise gene editing, raising both medical possibilities and ethical concerns. For exam questions, be prepared to explain how transcription factors, epigenetic changes, and the lac operon regulate gene expression. Use key biological terminology accurately to gain full marks. Thank you for watching.